Okay, if nobody want to ask questions, I for this section section, I want to throw out my maybe for the resistance breeding. The durable resistance is very important for the banana varieties. For the pathogen is keep evolving. Maybe some of them met this problem. The farmers grow the tolerant or resistant varieties in the field. After several years in the field, they found incidence of the TR4 increased significantly, which indicate that the pathogen maybe is evolving, or the pathogen have accommodated to the immunity step by step, 1 year by 1 year. And from I want to ask a question for the speakers. And from your research, how can we produce the durable resistant varieties to TR4? And James Down is still there? Maybe somebody has asked you this question. Maybe now you can conclude this question. Yeah, conclude it better now.
Sorry, what question was that? Can you just
And from your research, I wonder from your research, how can you produce a durable resistant varieties to TR4?
I think there are two ways to go. One is to stack resistance genes. I think one of the interesting things is that you know, there've been a number of presentations talking about resistance. We really don't. In most instances, we have no idea how many genes are controlling that resistance. In our case, we know that the transgenic lines we produced is a single gene. It may well be that many of the conventionally bred or selected cultivars also a single gene. I think one of the important things is for us now to go and look for additional resistance genes, different to RGA2, etc. So I think, yeah, we do want to stack genes. There is no evidence, at least at the moment, that a single gene for FAC resistance is going to break down quickly. So I think it's a good starting point. I would like to make one comment, and there've been comments about 218 in our trial and also in the other Northern Territory field trial. One of the things that we see most of the time is that field trials are concluded sometimes after the plant crop, but also after the first return. The results for both of our trials, we've gone to the second return, and in some instances the third return. And what we do see is that you find that cultivars or lines that are resistant or have low levels of infection plant crop start to have a higher level in the first return, but even higher in the second return. And considering most farmers grow their bananas for 3, 4, 5 returns, we really have to look at long-term field trials, not just plant crop plus 1 return. I think we're going to go further than that to find really durable resistance.
Okay, yeah, Dr. James, and in your presentation, for the transgenic line B, after 2 years of growing in your trial field, the infection rate is still 0.
Yes.
And compared, under the control reached to the after 2 years reached to 52%.
That's right. Yes.
Yeah, and for the severity of this disease compared to the in China, maybe I think the field the severity is not a severe your trial field is not very serious. And would you please consider test your the line B in the other places, in the more severe places?
Yeah, we're certainly considering that at the moment. One of the things we have to do is to fully characterize that line, and we may well take that line through deregulation, because these are GM lines, so we can't move them around as easily as we can with a conventionally bred line. But certainly our plan is to collaborate with others to screen these lines in different environments to make sure that they provide, or particularly line B, that it provides the same level of resistance that we're seeing in the Northern Territory in quite extreme conditions compared to other areas where in very different environments. So that's a very important thing to do.
Okay, thank you. Yeah. And Dr. Uma, are you still there? Uma?
Yes.
Oh,
Yes, yes.
thank you. And for your works, you screen the resistant varieties from the tissue culture virons from the crossing, and also from the evaluated the germplasm, also do the mutation breeding, and a very comprehensive works.
Thank you.
Also for this question, yeah, if you produce a durable resistant by varieties to TR4 by your research, how can you make it? Yeah, make a durable resistant variety.
No, right, like James already mentioned, as far as we know, it is it's governed by a single dominant gene. So breeding for that particular gene alone is always has a point of threat of breaking down in the due course of time with the evolving of new virulent strains. So in that case, definitely developing horizontal resistance is much more important. One thing we talked about is different R genes, which James was talking about. He has a set of R genes and NRCB has a different R genes. So we have to find out a couple of R genes which can be stacked and can be developed a new variety. The other one is also that I was talking about in conventional breeding. There are different sources of resistance. Those diploids with we do not know actually how the resistance is. First, you have to see the genetics, and once you understand the genetics, once you understand the genetics of inheritance, then definitely breeding a variety with resistance can give much more dividends for this kind of breeding work, and having a resistance for a different spectrum of pathogens.
Okay. Okay, thank you so much. Yeah.
Thank you.
Dr. Osheng, are you there?
Yes.
Okay.
Yes, I'm here.
In China, yeah, China mainland, in the past there is a very famous variety we call the Nunca #1. It's very in 10 years before. It's a tolerant or resistant TR4. It's very popular, adopted, accepted by the farmers, many farmers, and nowadays it's nearly disappeared. And from this variety, we know except the population size of the pathogen TR4 in the soil increasing 1 year by 1 year. And also, we can see from the resequencing data from a year, the pathogen is really involving many indels and SMP. So I think also relative to the evolution of the pathogen. So now the Nunca #1 nearly disappear. So it's indeed the pathogen is evolving or is overcoming the immunity of this variety. So from your research, yeah, you do the crossing. Maybe you can make one resistant variety. Maybe in someday will also be overcome by the TR4. How can make a durable resistance by crossing?
I think this question could be solved by 2 ways. Maybe the one way is for continue selection from the summer clone variation. We need to select year and years to find the good one which has good resistance in the field. We need to continue for selection in the field for 4 year and a year. This is not stopped. And another way is to for the crossbreeding. For the crossbreeding, it's need for many years. Maybe it has 20 or 30 years. I'm not sure for everyone if you could see big news in the internet. The CIAT in France, the CIAT released a hybrid is called the Cavendish-like. The hybrid is commercialized in the markets. In the in the international markets. It's a very good news for the crossbreeding breeds for breeders. So I think the crossbreeding could be possible to solve the TR4 problem. So for crossing and for the summer clone selection is 2 ways to solve this problem. So for durable for birds that get durable resistance to TR4, I think we need to have these two ways to solve the problem. Okay.
Okay, thank you so much. Yeah. And Dr. Mintoff, do you have any idea about this question?
About this pathogen? So what was the question?
And yeah, for the you do the many the trial field trials about the TR4 resistant varieties, selecting the TR4 resistant varieties. Do you have any idea about the making the durable resistance to TR4? Maybe can collaborating with some cultivation measures.
It depends on what the intent is. If you just want to screen for what's already out there, and I think Professor Dow is right in terms of the longer-term screening trials to make sure the resistance lasts. If you're talking about breeding, I think it's actually very important to do the screening initially for the breeding, the parental lines, because you actually can sort of use those lines to know what's resistant, and then you can sort of use that to sort of come up with hybrids later on. And hopefully you can have sort of that sustained resistance sort of move through to the into the new progeny.
Okay, yeah.